Thank you very much. It's a real pleasure to be here. I'm grateful for the opportunity to speak with you today and to review some of the work that's been going on at Rochester and elsewhere in adaptive optics over the last decade and a half. While most of my talk is going to be about imaging single cells of the living eye and functional imaging at that, I'll also say something about vision correction following on Chris's lead in his earlier talk today. Some commercial relationships to acknowledge. Listed here, I won't go through them, but I have them. And I also want to, Chris did a really, as he always does, did a great job of explaining adaptive optics. And I've got a slightly different take on it, and I hope you'll indulge me if it's a bit repetitive with this talk, but it took us so long to generate these animations that I couldn't resist showing them to you, so you're going to have to indulge me whether you like it. If you had a perfect eye, and you put a small pencil of light into that eye, the light would scatter off the retina and return through the cornea and lens, and you get this wonderful planar wavefront emerging from the eye's optics. But none of us have a perfect eye. Instead, the situation is more like this, where you put that same pencil of light in, the light comes back out, and as soon as it hits the back surface of the lens, then propagates through the rest of the eye's optics, your planar wavefront gets corrupted like this. Oh, thank you. Excellent. All right, so here's this warped wavefront that Chris was telling you about, and in this cartoon, I've exaggerated by a factor of 1,000 in the z-axis here. So, in fact, the distortions in the wavefront that we're really talking about are on the order of a micron or less, not this horrible distortion you see here. But in order to show what it is, I need to do that for the purposes of illustration. Now, you can take that wave aberration, and as Chris said, you can decompose it into Zernike polynomials. Zernike polynomials are really just nothing more than a description of individual aberrations that make up the total wave aberration of the eye that is that yellow warped wavefront I showed you in the previous slide. And there are, in this pyramid, this is just a means for a sort of taxonomy of aberrations. And defocus and astigmatism, of course, are the things that glasses can correct. But then there are these host of other aberrations, including many that aren't shown here. The pyramid keeps on going down below the screen. And these higher order aberrations are not addressed by spectacles, but can be addressed with adaptive optics. Everybody has a different pattern of aberrations, and so it's important and critical, really, if you want to correct them, to measure them in each person's eye and then correct them accordingly. Now, the history of measuring the aberrations of the eye is long and dates back to Shiner, even in the 1600s. But I'll pick it up in 1961, where a Russian named Smirnov did the first really provided the first really credible description of the eye's wave aberration. And interestingly, he said in 1961 that the method applied in the present work of determining the wave aberration is quite laborious. Although the measurements can be taken in 1 to 2 hours, the calculations take 10 to 12 hours. Therefore, it is unlikely that such detailed measurements will ever be adopted by practitioner ophthalmologists. Well, what Smirnov, of course, couldn't see was the advent of the digital revolution and our ability to do computations in unprecedented speeds. And so now it's possible to measure and even correct the wave aberration at video rates. So this is I put this in because it's a lesson to those of you who are learning to write papers. Never say never in your papers. But it's a beautiful paper, and I highly recommend reading it. Okay, well, how do we make this measurement of the wave aberration? Well, as Chris told you, you use a wavefront sensor. And if you had a perfect eye and you had that nice, beautiful, flat wavefront coming through, this lens that dices that lenslet array dices the wavefront up into a lot of separate beams that get focused on the CCD array back here, and they're beautifully spaced. They're nicely regularly spaced in that case. But in the case of an aberrated wavefront, those beams go off in peculiar directions, and you get an irregular pattern here. And all you need to do is, with your computer, is to measure the locations of these spots. And from those locations, relative to that perfect array I showed you in the previous image, you can compute what the shape of the wave aberration is. So that's basically how a wavefront sensor works, but it uses more lenslets than I'm showing here just for simplicity. So that's a Shack-Hartman wavefront sensor. Well, once we've measured the aberration, how do you then correct it? Well, as Chris said, you use a deformable mirror. Here's an example of one. And you make the shape of that mirror exactly the same, ideally, as the shape of the wave aberration, but with half the amplitude. And that works out to flatten the wavefront so that you get a nice planar wavefront emerging from the mirror. And you've now not only measured with a wavefront sensor, but corrected with your warped deformable mirror. So putting all these components together, here's your eyeball, here's your deformable mirror, here's your wavefront sensor. You've got a light source to deliver light to the eye, and then you've got a sensor, a light detector back here for recording your image, and here's a computer for doing your AO control loop. So you put a wavefront into the eye, it reflects off the mirror, and initially it's warped, it's not doing so well, but the computer updates the mirror status, changes its shape, so the next time the wavefront comes through, it's better. And you do this multiple times. You iterate until you get a nice planar wavefront emerging from the mirror, and that takes about a 1/30th of a second typically. Then you put an imaging beam through and let it pass to to your light sensor, which could be a CCD array or it could be a point detector, and that allows you to collect a high resolution image of the retina, as you see here. Now, an important feature of wavefront sensors, or of adaptive optics systems, that is nice for us, is that you can replace this light sensor shown here, you can replace that with a light source if you want to. And if you then have a point source emerge from this location here and let it pass through the eye, you might think, uh-oh, well, we've got a warped wavefront here, that's no good. But in fact, it's warped in just the right way so that when it goes into the eye, you get a nice correction of that wavefront, and you can produce a very sharp point source image on the retina using adaptive optics. So you can use it in reverse, and because of the reversibility of optics, the correction of the wave aberration for light coming out of the eye is identical to the one going into the eye, so you can do either vision experiments, as Chris described, or you can do retinal imaging experiments. So I want us to take a few minutes to say a few words about the vision correction, kinds of vision correction experiments that have been done using this technology. And then we'll get on to retinal imaging, which is the main purpose of my talk. As you heard earlier today, one of the first applications of wavefront technology was wavefront-guided refractive surgery. And truth be known, when I first started working in laser refractive surgery, all of my colleagues at the University of Rochester and vision scientists elsewhere, I think all around the world, that knew I was doing this, raised their eyebrows. And there was a lot of skepticism about whether this was ever going to come to any good and whether I was just selling out to commercial interest. And I'm happy to say that I think it did do some very valuable things, this marrying of wavefront technology to refractive surgery. And it is true, it was remarkable how fast the commercial interest in wavefront sensors grabbed onto this, and companies very quickly, within a very small number of years, embrace this technology, and so that nowadays most refractive surgery is done with wavefront sensors as a method for measuring the wave aberration. So the main advantage of combining wavefront sensing with laser refractive surgery was that there are aberrations produced by the procedure that were not characterized. Outcomes were not great. Patients were complaining of halos. They still do to some extent. But with a wavefront sensor, we actually had the ground truth about what surgeons were really doing to the eye when they did laser refractive surgery. And we were able to measure, for example, an increase in spherical aberration, which is produced by the surgical procedure. It's iatrogenic in that sense. And by measuring how much spherical aberration is produced under different conditions, it was possible to back that out, adjust the algorithm used to deliver the ablating spots in refractive surgery and correct for that spherical aberration, which improved the outcome of the procedure. So I think the benefit of wavefront-guided refractive surgery or customized refractive surgery isn't so much that it corrected higher order aberrations thoroughly and completely. We're still not there yet. But what it did do was provide much better outcomes for patients who were just getting conventional correction of defocus and astigmatism. Because now we had a way, which we never had before, of actually relating what the surgeon was doing to the actual optics of the eye in a very thorough and comprehensive manner. Another application of wavefront technology that especially Gunyang Yoon has been pursuing, although other investigators around the world have been interested in this as well. But Yoon at Rochester has developed customized contact lenses to reduce higher order aberrations. Now, in Chris's talk, he told you, and I agree with him, that for a normal eye, correcting higher order aberrations is a bit of a boondoggle. And the reason for that, as Chris said, is because those aberrations are relatively small to begin with. However, I wouldn't want you leaving this room thinking that all correction of higher order aberrations is a worthless exercise, because there are some cases where it's really quite important. And the example I'll use is this one, a keratoconic eye. Someone who has keratoconus has a cone-shaped cornea. A cone-shaped cornea has higher order aberrations that conventional spectacles cannot correct. So there's a huge benefit in some cases of correcting the higher order aberrations in an eye like that. Keratoconic eyes can be legally blind in the US because of their, throughout the world I suppose, because of their deficiency in the shape of their cornea. And with contact lenses, such as the ones that Yoon has developed, you can actually greatly reduce the higher order aberrations, a 3 times reduction in the higher order RMS in this particular eye, for example, which can take a patient to from 20/100 to 20/40 or something like that. Not perfect vision, but a vast improvement, multi-line, 2-line improvement in their vision, which is significant for them and the quality of their life. So correcting higher order aberrations can be very useful depending on the kind of higher order aberrations you have. And just as some people have astigmatism and benefit from having it corrected, other people, for various reasons, not just keratoconus, have higher order aberrations that warrant correcting. Not to produce supervision, but to get them closer to normal vision, which you can't do with spectacles in that case. Now, another application of this technology that I've been talking about for seems like decades, it couldn't have been that long, a decade and a half probably, is that ultimately, I think that adaptive optics, if it can be made cheaply enough, will replace the current 4-opter. This device, which I had to talk about this because I'm in an optometry school. So this device is a relatively inexpensive device compared with an adaptive optic system, but it's a kind of a kludge. It has 88 separate lenses that you slide in and out to refract the eye, and you've got that laborious procedure that those of you who do this sort of thing are very familiar with, of asking which do you like better, A or B, and you can't always count on your patient to deliver a reliable response there. So it's a pretty inaccurate procedure, if you don't mind my saying so. But with adaptive optics, you really have a complete description of the monochromatic aberrations of the eye. And if you want to build in other things to your model of what the best refraction ought to be, forget about correcting higher order aberrations. Let's just suppose you're prescribing glasses. You have a very good shot at getting that right with a wavefront sensor, and then the role of the mirror in that case is just to show the patient the outcome of the refraction that you've prescribed for them. So in a small fraction of a second, once the patient is aligned in the system, you could have their prescription and have an accurate one. Now, when I tell this to optometrists, they jump back in horror because they know that their bread and butter depends on asking patients over and over again, which do you like better, A and B, and they're trained to do that, and there's just this inertia that's very difficult to get around. So it may be a while before this technology gets adopted, but I think it will be adopted ultimately as these systems come down in price. And one of the things that the optometrist who's sort of a dyed-in-the-wool practitioner will say as well, this is an objective method, and what about the brain? I mean, the brain is this mysterious, unknown thing, and we've got to pay homage to the brain, and that's why I do what I do in refracting my patients so carefully subjectively, because you need that component. But in fact, in recent years, we've learned a lot about how the brain responds to patterns that are defocused, and I'll give you just a little hint of that, which is another example of how adaptive optics can be used to learn about vision. And this gets to the point that Chris made about the importance of adaptive optics and vision testing. So in collaboration with Pablo Artal at the University of Murcia and his colleagues, a number of years ago, we did an experiment in which we took normal subjects and we showed them their own a target, letter target, doesn't matter, visual target. They were able to look through an adaptive optic system at a target, and we showed them that target with their normal aberrations, and then we were able to use the adaptive optic system to create a situation where their aberrations were rotated. So we weren't doing anything to the absolute amount of any aberrations in the eye. We were just rotating them. And then we asked the subjects we made measurements really, but I won't go into the details of how we did this, but we made measurements of the image sharpness as a function of the angle of rotation. And don't pay attention to the small deviations here. I don't think they're meaningful. But the point is that all rotations away from the one that the patient was accustomed to produced a marked reduction in the sharpness of vision. So what this is showing is that because we did the experiment in such a way that the aberrations weren't really changed in magnitude, just rotated, this shows that your visual system is adapted, the nervous system is adapted to your own pattern of aberrations. And those of you who are practicing optometrists won't be surprised about this, because you know that you undercorrect astigmatism routinely because your patient squirms and complains and comes back and returns the spectacles a week later when you try to do a full correction and someone has a large amount of astigmatism. So I think you have a rule even for this. I've forgotten the name of it, but you correct, 75% or something of the astigmatism in patients that have large amounts. Well, we now know that if you wait and give patients a time to acclimate to that a new wave aberration, say one that's reduced the higher order aberrations to 0, their visual performance will actually steadily increase. So initially, you don't get much psychophysical benefit from correcting higher order aberrations, or you get some, but not as much as you might expect. But that grows as the nervous system learns to see through this new pattern optical image that's available to it. So even if you initially, even the correction of all the higher order aberrations doesn't produce the best image quality, because your nervous system has a lag. It has to take time to capitalize on plasticity and take advantage of that improvement. Now, I just wanted to finish this section on laser vision correction, or on vision correction, by telling you describing an example of some recent work going on at the University of Rochester that I'm not involved in, but except peripherally, really, done by Wayne Knox and Crystal Huxland, and that I'm very excited about, because laser refractive surgery, one of the big problems with that, of course, is you have to cut this flap on the cornea. Ouch. And then you go and you zap tissue. You're removing tissue always to do the correction, right? So there are a finite number of times, like maybe once, that you really can do that. You can afford to thin the cornea that much by removing tissue. But Wayne and Crystal have discovered that there is a regime between very low light levels of illumination with a femtosecond laser and very high light levels. Low light levels do nothing to the cornea. High light levels damage the cornea and ultimately will remove tissue a la conventional laser refractive surgery or LASIK. But there's an intermediate regime where you can change the refractive index without appreciably increasing scattering or damaging the tissue. And this is illustrated here. This is actually done in plastic, or maybe this is a hydrogel. I'm afraid I've forgotten which. But the point is that if you look at this material, man-made material in bright field illumination on a microscope, you see very little, if any, structure at all. Because all you've done is change the refractive index. You haven't changed the absorption properties of the material or scattering properties. You've just you've written a diffraction grading, and you can see that here. So if you image with phase contrast, which picks up refractive index changes, there's actually a pattern written into that material. And the really nice thing is that that can be applied to a living cornea as well. It's been done only so far in the cat, but they have succeeded in producing changes in the refractive state of cat eyes approaching 2.5 diopters. That's what they, based on their measurements of us, they predict they should be able to do that. You'd like to have it more than that, but still that's an exciting development. And they think it will take about 15 minutes in a human eye to realize a correction of that kind. The other exciting thing is that it's been stable. Whatever refractive changes they implement have been stable over a period of 9 months. So I think there's a possibility here that we may be entering an era sometime in the relatively near future. A lot more work to be done on this. It's not ready for use in humans yet. But we may be entering a time when LASIK, as we know it, disappears, and it's possible to introduce, at least in people that have low refractive errors, to actually do that correction without removing tissue by taking advantage of this refractive index change that hadn't been discovered before because you couldn't see it. You know, you couldn't. You can when you blast a piece of cornea and you see it go opaque, you know you've done something. If it just changes the refractive index, you need a special technology even to tell it you've done that. So we're very excited about this technology, and as I say, you know, there are other things that need to be worked out before this is really ready for human use. But I think it's exciting and has great potential. Really could revolutionize vision, because if you could do this over a large range of refractive errors and correct astigmatism and sphere, so many of us could throw these away, and that would be exciting without having to remove tissue or cut a flap through your cornea. But we're not there yet. Okay, I want to now move on to human imaging and some of the work that's been going on there at Rochester and elsewhere. So just to give you a sense of the spatial scale at which we're operating when we take pictures of the retina with adaptive optics. Here's an eyeball, and here's a 10 millimeter square on the exterior of the eye. If we project that square in the interior, this is sort of, roughly speaking, order of magnitude what a conventional fundus image would be like. But with adaptive optics, we're going nearly 2 orders of magnitude smaller than that. Here's now a square that's 100 microns on a side. And this is the kind of view that you have of the retina using a conventional camera, a fundus camera, for taking a picture of the inside of the eye. And you can see there's almost no structure visible at all. Because of the, largely but not exclusively because of the aberrations that exist in the eye when these pictures are taken. But with adaptive optics, looking at an area of comparable size in a living human patient, you can see this kind of detail, where you can see cones. These large cells are the cone photoreceptors. The rods you can see in between. The rods are 2 microns in diameter, which is close to the resolution limit of currently available adaptive optic systems. And I do have to give credit where credit is due. So Alf Dubra, who was a student of Chris's, and then I was privileged to have him for a few years, and now has moved on to the Medical College of Wisconsin. He's right now sort of the premier guy at tuning up adaptive optic scanning laser ophthalmoscopes, and this is his image that he acquired with one of his instruments. And we're using at Rochester his designs now, and no doubt we'll be using many of his tricks that he developed there going forward. Okay, so this is a bit dated, this material, but I want to take you through some of the work we've been doing in color vision, in part because there's some recent developments that I'll tell you about at the end that I think are a real technical tour de force done by a different laboratory independently of us that I think will make, at least I'm excited about this again after spending many years trying to achieve certain results that I'll talk about next. But you know, Thomas Young was brilliant, right? He knew, he helped decipher the Rosetta Stone. He was responsible for key experiments on interference phenomena and the wave nature of light, and he knew an awful lot about the optics of the eye. He also was the first person to clearly articulate that you have 3 fundamental channels in the visual system responsible for color vision. And I wanted to show you, some of you may have seen this before, and I apologize if you have, but some of you may be familiar with this, the reviews that he was receiving on papers around that time. And I recommend this as entertaining reading. You can go to Henry Brome's original reviews. We now know who that anonymous reviewer was. It was Henry Brome, an Englishman. I don't think he was from Wales, so don't worry. But he said things like, this paper contains nothing which deserves the name either of experiment or of discovery, and as it is in fact destitute of every species of merit, blah, blah, blah. And you can read this one. It doesn't get any better when you read this on your own. So one thing we can conclude from this is that peer review was alive and well in 1802. And the other reason I like to show this is because, especially for the students in the audience, because you know it's so demoralizing when that negative review comes in. And so what I recommend to each and every one of you is you take these quotes and you put them, as I have done, and you put them above your computer so you can read them. And when those negative reviews come in, you read them and you realize then every negative review is just more evidence that you're an unrecognized genius like Thomas Young. Okay, so Thomas had this brilliant insight that there were three kinds of channels. He didn't know that they were cones in the human eye. And since then, we've made remarkable progress in understanding the spectra of these. There's little debate now about exactly what those spectra are, even though when I started as a graduate student, these were largely unknown, and a lot of controversy about what shapes they were. We knew even less at that point about the topography of the three kinds of cones. And so as Chris pointed out, early on, Austin Rorta, shown here, largely single-handedly, because I was on sabbatical at the time, working with Dennis Dacey at the University of Washington. But we had phone calls daily, practically, as I was charting his progress on these experiments. And he was able to, using the method Chris described, to categorize the three kinds of cones in the human eye. And that opened a whole series of experiments that weren't possible before, because now we knew that the S-cones were, well, we'd already known that, I guess. The S-cones were, at that time, were rare in the retina, accounting for on the order of about 6% of the cone population. What we hadn't known was the relative numbers and packing geometry of the L and M cones, the long and middle wavelength cones shown in red and green here. Those turn out to be randomly organized, and we were able to provide direct evidence that their relative numbers changed dramatically in the normal population. But before I show you that, I did want to mention that we also have been using this technology, thanks to Joe Carroll, who was a postdoc in the lab many years ago, to look at diseases of the eye, especially diseases for which we have the genetic basis for the disease. So now we can look at the genotype, for example, for a kind of color blindness, and we can compare that with the phenotype that we can image in the living eye for the first time. It was very difficult to know what the phenotype might be of a person with a particular mutation, just because you couldn't count on them to die in your laboratory and give up their retina in time for you to get it in a microscope and actually image it. So with adaptive optics, we're able to see that. And here's a case of a normal trichromat, which has a rather complete mosaic. You can ignore the variations in reflectance, which are found in all normal eyes and are perfectly normal. But note the difference with this particular deuteranope. This is someone who's colorblind with a version characterized by deuteranopia. And you can see holes in the retina. These are cones that aren't reflecting light back properly. And they are probably not dead cones, but the outer segment is probably not functioning. This mutation is a mutation in the genes that produce the M pigment, and that's responsible for these microscotoma that pepper the retina that you see here. And so I won't take you much further down the road of clinical work with adaptive optics in today's lecture, but suffice it to say that that we're having a lot of fun looking at a lot of retinal diseases in a novel way now, because we can image them non-invasively in human eyes in a way that was at a microscopic scale, which was never accessible really in practical terms before, and certainly not in a living eye. So as I mentioned before, the ratio of middle to long wavelength cones is highly variable in the population. And I'm showing you data now for normal eyes. These are not colorblind eyes. These are eyes like we would have represented here and screened against to remove those who are colorblind. And a remarkable variation with this eye up here having 3.5 times more M cones than L, and down here, this eye has 16 times more L cones than M. So a huge variation, about a 40-fold range in the ratio of L to M cones in the normal population of eyes. Incidentally, people often ask about these gray regions. Those are just regions where we generally underneath blood single capillaries, where we decline to classify the cones because well, in Chris's terms, we didn't want to take artistic license with those particular photoreceptors. So we did not classify those, but the rest of them are classified with an accuracy on the order of 5% or better in every case. Okay, you might ask, well, okay, how different is the color vision going to be across these different individuals? You might think, oh, the world's going to look really green to this individual and really red to that one. But if you give each of these subjects a monochromator and allow them to adjust a knob to control the wavelength that looks perfectly yellow, neither reddish nor greenish, which is thought to be a function which is known to be a function that depends heavily on the middle and long wavelength cones, not necessarily their number, but on those cone photopigments, what's this so-called unique yellow point. You find that across a very broad range of LDEM ratio, in this case computed from ERG data, you can see that there's almost no variation in unique yellow. So these very red-rich, or L-cone-rich and M-cone-rich people all agree within a couple of nanometers on what's perfectly yellow, despite that huge difference in their retinal mosaics. So we now know from experiments I don't have time to describe that that there are plastic mechanisms that set what unique yellow is, and there's a good reason why the visual system is organized that way for defining the boundaries and color experience. And it has little, if anything, to do with the hardware at the front end, at least with the relative numbers of cones for large-scale stimuli. But the next question we ask, well, what about small stimuli? How do, given that you have a mosaics that, I'll go back, that look like this, you know, one of the deficiencies of this kind of a design for a retina is that you only got one spectral sample at every location in space. What you'd like to have is you'd like to have 3 spectral samples at every location in space, right? Because then you get a value, an estimate of the color at every location in space, but instead you have a single tiered retina with one plane of photoreceptors in it, and so that necessitates having only one kind of cone at every location, and that creates a problem for vision, the problem being chromatic aliasing, and I'll give you an example of that shortly. So early on, David Brewster noticed that if you look at very fine patterns and etchings, like the waves that are, the fine lines that are used to represent the sea in maps of the maps of the world, you saw kind of pastel colors that would come and go. And maybe some of you have seen this on your computer screen, looking at a white portion of the screen. Maybe you've seen some pastel hues there. We later showed that that was consistent quantitatively with chromatic aliasing. And here's an example in digital imaging with a camera, not with the eye, of what chromatic aliasing is like in a bad case, where this suit looks like it's covered in garish stripes because of undersampling by the 3 kinds of sensors in the CCD array used to take this image. And then later, following David Brewster's work, a Swede named Holmgren, who's a physiologist, noticed that stars tend to, very small point sources focused on the retina, tend to fluctuate in color over time. And he attributed that to the granularity of the photoreceptor mosaic and having 3 kinds of cones. And so since then, many people have studied this phenomenon in many ways. And the expectation that most people held about that experiment, what would happen if you stimulated one cone at a time, is what I'm going to call, it's sort of a straw man theory here, but I'm going to set it up anyway. The elemental sensation theory is what I call it. And a good articulation of that theory comes from the work of Hermann von Helmholtz, who said in 1866 that the eye is provided with 3 distinct sets of nervous fibers. Stimulation of the first excites the sensation of red, stimulation of the second, the sensation of green, and stimulation of the third, the sensation of violet. So Helmholtz's view, and nearly everybody after him, thought, okay, 3 kinds of cones, each one has its own perceptual experience that's locked to that cone type. And if you were to go and stimulate an L cone at different locations in the retina, every time you get the same color experience. In Helmholtz's view, you get red. Now, some of you may say, oh, well, we know that there's an opponent color theory that is sort of piggybacking on top of the trichromatic theory of vision. We know that there are interactions between cones, but still that doesn't really violate the fundamental premise of the opponent elemental sensation theory, because even in the case of opponent interactions, illustrated here very simply and schematically, where you know, you take the difference between different cone classes. If you want to compute blueness, for example, you subtract the S-cone signal from the sum of the M and L. We don't need to get into the details of this. The point is simply that even with an opponent colors theory of color vision, you have this mapping, this one-to-one mapping between single cone stimulation and color experience. The opponent model might make different predictions about exactly what color you would see, but still every cone would generate the same experience. Well, so this led, with the advent of adaptive optics, we were able to try this experiment, and Heidi Hofer really pioneered that work in my laboratory as a graduate student. And adaptive optics makes it so much better because by focusing a point source onto the retina, you can, with adaptive optics, correcting the aberrations, you can get a light distribution that is something like this. We don't know exactly what the light distribution is, but this is a quantitative estimate of what it probably is like with adaptive optics. Without adaptive optics, the light is spread over a much larger area, and you're much less effective at stimulating a single cone one at a time. So with adaptive optics, we were able to greatly increase this rather subtle, ephemeral color fluctuation that Holmgren had reported, and we were able to start making color naming measurements of what these flashes of light look like. And we always used, well, not always, but in the experiments I'm going to tell you about, we used a single monochromatic light at 550 nanometers, if memory serves. So there was only one color presented to the retina. And we just asked people to report what they saw to multiple flashes. The flash was set very near threshold. And this is pretty complicated, but let me take you through it and sort of highlight the important parts that you need to understand to grasp the remainder of this portion of the talk. We did this in 5 subjects, and the 5 subjects spanned the range in the normal population between the M-rich, so that was the guy at the very upper left part of that arrangement of mosaics I showed you earlier, all the way to the most L-rich subject we had in our population. So we did this psychophysical work on the same eyes whose mosaics we had characterized with this retinal densitometry AO method. And what we noticed in all the subjects, no matter what their L to M cone ratio was, is that they needed a lot more than just 3 color names to describe their percepts. They did not, in fact, we started with 3 names, red, green, and blue, or red, green, and yellow, actually, believe we started with. And they just said, no, you can't do this. I see blue, and yet we were using a light that was chosen not to directly excite the short wavelength cones that had been thought to generate the experience of blueness alone. And yet everybody reported some blue sensations, as you can see here. These bars indicate the colors that they were reporting. So that was one interesting thing, is without stimulating the short wavelength cones, people were reporting blue experiences. The other thing that was peculiar was that especially everybody reported white experiences, but especially the people that had very extreme ratios, either M-rich or L-rich, saw lots of whites. Well, how do we explain these results? Well, they're inconsistent with the elemental sensation theory as Helmholtz, he wouldn't have called it that, but as his quote sort of articulates. And so we went looking for another theory, and Dave Brainerd from the University of Pennsylvania stepped up to the plate and generated a theory for us, which I'm going to just qualitatively describe here briefly. I won't be able to go into great detail about it. But I think captures the essence of why we think it doesn't make sense that the retina should be, or the brain even should be organized according to the elemental sensation theory. It actually makes a lot more sense that you would get a rich variety of color names when you stimulate one cone at a time. And the essence of this idea comes from Bayesian theory. So the idea is that what the brain is really supposed to be doing with the retinal image is making the best estimate it can based on the photon catches it gets from the photoreceptors of what the external stimulus is there, right? So you'd like the brain, ideally the brain would be optimized in some optimal way to make the best guess it possibly can for every cone that might be stimulated in your experiment. Now, keep in mind that the brain has little or no experience, really no experience with spots as small as the ones we can deliver with adaptive optics. So the brain is used to making its decisions about what's out there based on combining signals from multiple cone classes, okay? So let me go right to Dave's Bayesian model to give you the flavor of that. So the idea is that suppose that you had a mosaic that had only, in a local vicinity, only L cones. And you stimulated this cone right here, shown with a little white spot. Now, this is not to say that this is a a colorblind retina, it's just locally colorblind. There might be M cones elsewhere, but locally there are only L cones. Well, if you stimulate that, that cone, you'll get a response, but the reconstruction this model comes up with is white, not red, because there's no comparison possible to what the M cones are seeing. So the best guess is, oh, just some achromatic thing. We don't know what it is, and the best guess for this Bayesian model is that it would be white. However, if we were to surround that same cone with a sea of M cones, now if you stimulate that single M cone, L cone, the brain knows, if you will, that the M cones in the vicinity are saying, I don't see anything. Now, what kind of stimulus out there in the world would produce a really strong L cone response with no or little M cone response around it? It would be a very saturated red stimulus, right? So our argument is that these color experiences vary all over the map because the brain is organized to do a more optimal thing about deciding what the stimulus is out there. And to make that optimal decision, it's taking into account the signals from adjacent receptors. Now, this theory is exactly that. It's a theory. It remains to be tested. And the good news is that Austin Rorta, completely independent of us now, at the University of Berkeley, has improved eye tracking capability so that he can actually deliver a spot to any cone repeatedly over time. In the Hofer experiment, we just relied on the statistics. We didn't control exactly which cone we stimulated. We just let fly with a bunch of spots and asked people what they saw, but we weren't able to repeatedly stimulate the same cone over again. And the good news is that Austin has learned how to track the eye so carefully that he can deliver a point source of light to a single photoreceptor over and over again over time without missing. Or if he does miss, on rare occasions, he knows he missed and he can throw that data point out. And so he's repeating all these experiments, and while I don't have any of his data to show you here, he did share it with me last week, and he hasn't violated the Brainerd model yet, let's put it that way. And he's getting consistent, when he stimulates one cone, he consistently gets the same color experience, and that color experience doesn't fall neatly into these three categories you might expect from the elemental sensation theory. So I wanted to say a little bit more about eye movements, because I know some of you are interested in eye movements, and it's absolutely critical to the future of high resolution imaging with adaptive optics, and has been one of the fundamental barriers to doing more science and more medicine with high resolution AO imaging, because the eye is moving over time and the signals that we can collect, even with our most sensitive devices and the most sensitive photodetectors, are photon starved. We really need to be able to average multiple frames together in order to get enough signal to figure out what's happening in a microscopic spatial scale. And most of the work that we do at Rochester with this technology relies heavily on frame averaging. But, of course, the eye is always in motion. Here's a very good fixating eye, and you can see how the photoreceptor mosaic is moving relative to this letter E projected on the retina. And this is actually work from, again, from Austin Rorta's lab at UC Berkeley. And you can also see some intra-frame distortion because the eye is moving during the acquisition of a frame. It's a scanned image, and so there's also motion there. And Austin has developed the technology now to the point where he can stabilize that letter E on the mosaic. You can see a little bit of jitter here and a little bit of distortion in the mosaic, but it's far more stable. And if you look carefully, I think you'll see that the jitter here is smaller than the diameter of a single cone photoreceptor. So we've taken Austin's lead, and working with Jean Yang, who's now at Rochester, we've further improved this capability. So here's a trace obtained with our latest instrument. This is one we're developing in collaboration with Canon, who is developing adaptive optic scanning laser ophthalmoscope prototypes, and we'll be selling them in the relatively near future, making them widely accessible in the relatively near future. And so the method we're using is a hybrid method. It has both an optical stabilization component to it, as well as a digital registration method, and I won't elaborate on the details of that. But here's uncorrected eye motion, as you might typically encounter in a patient in your in the clinic. This is probably a little better than the patients that that John sees, for example, that have nystagmus. It's a lot better than that. But with optical stabilization alone, we can drop the RMS error in position to 1.5 to 3 microns, which is on the order of the size of the smallest foveal cone at 2.5 microns, or a rod at 2. And then when we add to the optical stabilization a digital method that's also, and this is all done in real time, by the way, then we can drop it even further down to a level of about 0.4 to 0.5 microns, which is a quarter, a fifth the diameter of a foveal cone. So we're able to get down to the point where you really can average many frames together and build up the signal-to-noise ratio you need in order to get reliable data in very dim imaging situations. And I'll show you some of those situations, some of the more egregious ones in the next portion of my talk. But that was a key development, and I really would credit Austin primarily and his colleague Yang primarily with developing this capability. Well, another problem we have in retinal imaging is the cones, they just glow like crazy. They're beautiful, and the reason they glow so nicely is because they're waveguides, which means when light comes into a cone, it stays by and large in that cone, travels down the length of the cone, and reflects off of various surfaces inside the cone and back out toward the pupil, where we harvest the photons and make an image. So cones are like little spotlights shining back at you, little retroreflectors shining back at you, because of their waveguide properties. But the rest of the retina is depressingly uncooperative. And the inner retina in particular, which we'd very much like to image, the ganglion cell layer, for example, in glaucoma, we'd love to be able to do someday cell counts of ganglion cells as glaucoma progresses through the inner retina, and we just don't have that capability yet, but we're working toward that. That's one of the holy grails, I think, for this whole community of people who developed this technology. And the problem is, of course, that all this stuff really needs to be transparent. If it's not transparent, then the photoreceptor's performance will be compromised for the person that owns this retina, and that means that there's very little light coming back to see this with. Now, you may say, oh, well, wait a second, in OCT images, you can see this just fine, but OCT images are really the logarithm of the linear image that's acquired, and that's done deliberately to make these very weak structures more visible. And so the reflectance from the ganglion cell layer is on the order of only a 60th of the reflectance from the cones. So we'd like to find ways of enhancing the visibility of this layer. Now, you heard about two-photon imaging earlier today, and many of you are familiar with it, I'm sure. One of the really nice things about if you could use two-photon imaging in a living eye is that all the cells in the retina have mitochondria. Mitochondria have NADH in them. NADH is a fluorophore that you can image with two-photon imaging, and so cells that are transparent under normal illumination, when you do two-photon fluorescence imaging, all the cells are just really visible. So this is not adaptive optics, unfortunately, in the living eye. This is ex vivo imaging with a two-photon microscope. But look at the ganglion cells. They just stand out just beautifully. The nuclei here are quite dark. The photoreceptors also are very clear. And moreover, the signals that you get from these cells are functional signals, in the sense that the amount of signal that you get depends on the metabolic vitality of that particular cell when you're imaging it. So great potential if you could only figure out how to do this in living eyes. Well, Jennifer Hunter at Rochester has had a go at this, and one of the really nice things about two-photon imaging that's an advantage for imaging in the eye that you wouldn't hear about in talking to people who do two-photon imaging in cortex. The reason they like two-photon imaging is you can penetrate deeply into the cortex and collect a weak signal coming back out. But in the eye, there's this other cool advantage, which is that you can tickle molecules that are outside of the spectral window of the eye's optics. So, for example, here's the spectral window of the eye's optics. This is the ocular transmittance, and it's largely due to the cornea and lens and how much light they absorb. They really suck up all the light in the UV. Very difficult to get UV light onto the retina because of this limitation. NIDH, on the other hand, has an excitation spectra that falls right there outside the window. So how can you use two-photon imaging to study this fluorophore that's inaccessible using other methods? Well, with two-photon imaging, you put in infrared light. That goes through the window, so you can get light in. But when it gets to the retina, it pretends, so to speak, and I'm using that anthropomorphic word loosely here, but it behaves as though it's light of half the wavelength. So it behaves as though, because of this nonlinear interaction with the NADH molecules, it behaves as though it's in the UV, tickles the molecule, excites the molecule, and the fluorescence is at longer wavelengths. So it comes back through the window. So this band shows the fluorescence. So you can put light in, you can tickle a molecule you could normally not see at all, and then the light comes back out through your window, and you can learn about that molecule in the eye. So Jennifer capitalized on this by, in a monkey, building a two-photon adaptive optic scanning laser ophthalmoscope, and in the monkey retina was able to show this image of photoreceptors. Each little spot here is the inner segment of a cone. Inner segments are chock full of mitochondria. They have a very strong NADH signal because of that, and we think that's what we're imaging in this case. The problem is that this required 12 minutes of integration of light to get this single image. So we need to find ways of increasing the efficiency of this method if we're ever going to use it in humans to image, say, ganglion cells. The ganglion cell signal will be much weaker than the inner segments of cones. And we're working on that. We have a strategy for doing that, and we're working on it. But not making any promises at the moment. All right. Well, I want to now finish by telling you about some other work that we're doing that uses adaptive optics in another way in order to study the retina. And the scientific problem that we're interested in solving here is to understand what really is the signal or set of signals that the retina is conveying to the brain. You probably all know you have 1.2 million ganglion cells in a typical human eye, roughly speaking, and that may seem like a large number, but then remember that each ganglion cell is, roughly speaking, responsible for a different portion of the visual field. So let's call it 1.2 million pixels in round numbers. And yet, your cell phone camera is a 5 megapixel camera. So you have 5 times as many pixels in your cell phone as you have ganglion cells to capture the entire wide-angle field of view that you all enjoy with each of your eyeballs. And so how is it that this happens? And especially since these ganglion cells don't come in just one flavor, right? There are perhaps as many as 2 dozen different kinds of ganglion cells, each one sending a slightly different kind of information up to the brain. And we know something about the functional roles of these different ganglion cells, but far less than we'd like to. Most studied are the midget and parasol cells down here that become the magnocellular and parvocellular pathways in the LGN. They're very numerous in the retina, and many of my colleagues, and I'm thinking of Peter Lenny and Bob Shapley, are sort of M and P chauvinists. They think that, what I mean by that, I'm probably being slightly unfair to them, but I think if I went to Bob Shapley and asked him about this today, he would still tell me that all that we really care about for vision is the midgets and the parasols, and the rest of this stuff, this is all just fluff and completely uninteresting. I think a lot of retinal physiologists take a different view now. They think, oh, maybe these cells have their own unique function. Each cell class completely tiles the retina independently of the others. And as we do discover the functional roles of these cells, we're finding that they have very specific circuits for doing very specific jobs. And I'm excited about this because, my cortical colleagues are always dumping on me for studying this cold and distant outpost out there in the retina, and I'm always criticizing them for studying the extraocular ganglia, which they call the brain. But I think there's an interesting situation here in which it may well be that the retina is doing more processing than we thought, and relinquishing that, freeing up the brain from having to do some of the processing that we might have otherwise thought happened there. And an example is the recent relatively recent discovery of lots of motion sensors in the coming out of the mouse retina, and we don't know whether these exist in the primate. I have a suspicion that they do, but we don't have direct evidence of that. Another example would be the intrinsically photosensitive ganglion cells. If you'd asked me a decade ago, if you told me that there were ganglion cells that were sensitive to light on their own without input from rods and cones, I would have laughed, because I would have thought, well, we know what the light sensors are in the eye. But of course, as many of you know, there's been this discovery of these cells, and they do have important functions. Maybe not for the finest visual discriminations that people can make, but certainly having a cell class that controls the pupil diameter is an important thing, or another cell class of these cells that controls the circadian clock. So as we learn, as I say, as we learn more about these cells, these rare cells in the retina that aren't as popular as midgets and parasols, we are discovering that the retina really gets credit for a lot more than we might have originally thought it did. Why is it that we don't have this all figured out, you may be wondering? Why is it that we can't just go in and record from these cells and get the answer and then go home? And the answer is because the technology is deficient in a very fundamental way. So this is a picture from Dennis Dacey's laboratory, and it's the way he's been doing just wonderful, beautiful work on the organization of the retina, especially ganglion cell function for color vision and other things for many years. And he takes the retina and he cuts it out and he slices it up like this and puts it in a dish and does an admirable job of keeping it alive for about a day. And then he records from these ganglion cells one at a time with a single microelectrode. Now, when you talk to the people who do this, and they've done beautiful work, and I'm not dissing what they've done. I have ultimate, just incredible respect for Dennis. Spent a year on sabbatical with him to learn how he did the sort of work, the magic that he does. But imagine the problem. If you have a cell class that, say only 4,000 of those 1.2 million cells is a cell of a particular class, the chances of encountering one with a single microelectrode is small, and getting two, which might be what you'd want to write a paper about them, is even smaller. And so when you talk to these guys, what they'll tell you is, I saw the most incredible cell. It had these problems. Never seen another one like it again, but it was there. And so what we need really is a technology that allows us to record from many cells simultaneously. Now, really right now, the premier technology of that kind has been pioneered by E.J. Chichelnitsky, and you may be familiar with this beautiful work that Field and Chichelnitsky in particular are responsible for. And what they were able to do is using an array of electrodes, record from many cells simultaneously, and piece together the cone inputs to individual ganglion cells. So that's what you're seeing. These dots are the cones of all three flavors, and these lines show the ganglion cell connectivity between the cones and the ganglion cells. Beautiful, beautiful work. But it has its limitations too. One being that you can only sample the cells that your electrodes happen to be near. And the second being that you have to do this in an excised retina in a dish. So you don't have the opportunity of doing in vivo experiments that you could ultimately combine with behavioral methods. So we've been using adaptive optics to try to remedy that situation. And while I don't have any scientific results to show you, I do have some some preliminary results on how this technique is working. And it's an optical method. You won't be surprised. It's an imaging method in which instead of poking around with a microelectrode, we image all the ganglion cells in a region. And we do that by using a genetically encoded calcium indicator that gets ganglion cells to express a fluorophore that very similar to what you heard in Mateo's earlier talk, which allows these ganglion cells to respond in a way related to their activity. So the fluorescence that they generate increases with increased neural activity. And then we've been doing some of this in mouse and some of it in primate, and I did want to put in a plug for the mouse, which I think there's been a lot of mouse prejudice around that I'm trying to dispel. Melissa Gang showed that the mouse optics, which we always thought were, and the literature says, is horrible. Turns out the mouse has optical quality as good as a human eye if you just measure it in the right way. And with that, she was able to image a number of different ganglion cell types, which you see here in a living mouse eye, labeled with, in this case, just GFP, not that calcium indicator I'm going to get to in a moment. But she did this in a mouse. We've also been doing similar kinds of experiments, imaging ganglion cells in vivo in living animals with intact eyes in monkey as well as mouse. Now, Bill Merrigan and Lu Yin at Rochester have been using that calcium indicator, GCAMP, and getting that expressed in ganglion cells. So here you can see about 80 ganglion cells that express different levels of this calcium indicator fluorophore. And this slide shows you, if you look at just a few of these labeled, and then you see the traces over here, when you deliver a light flash to the retina, you can get these ganglion cells to increase, or in at least one case here, decrease its firing rate optically. So now we have an optical way of looking not at just the cells a microelectrode array might happen to hit, but all the cells in the field of view that are labeled with this fluorophore. And we can then increase, we hope, the number of neurons we can sample. We can go on this grand fishing expedition where we decide what we think the retina might be capable of doing, whether it's motion perception. Maybe there is some orientation selectivity, we don't know. There is in some animals, whether there are cells that are sensitive to focus is an unknown issue at the moment. And we think we can answer some of these questions by using this kind of a method. We haven't deployed it yet for that purpose, but those experiments will be coming up shortly. Now, finally, I wanted to mention that and remind you that this same optogenetics related approach for recording from cells also has the potential to stimulate cells. And you heard a little bit about the use of channel rhodopsin earlier. One interesting alternative to this approach to vision restoration in the blind, this approach is the second sight approach, which has gotten a lot of press. But it's a pretty kludgy approach in many ways. It requires a camera external to the eye, and it requires a difficult surgery to implant an array of electrodes into the retina. It depends on the continued efficacy of those electrodes over time, and experiments of this kind are notorious for loss of connectivity between the electrode and the neurons you're trying to stimulate over time. The number of pixels that you can create, the number is dependent on the number of electrodes, which is generally fairly low. So we're very interested in the possibility, and others certainly are doing experiments of this kind quite successfully in mice, at least demonstrating that you can put channel rhodopsin in mouse bipolar cells, and you can get these mice to solve mazes where the solution involves detecting the location of a light source. So we've been able to get the same molecule now expressed in monkey fovea, and near this ring shows you the ganglion cells around the center of the fovea that are expressing channel rhodopsin, and we're excited about the possibility that you might be able to bypass the photoreceptor mosaic, if that's the mosaic that's been compromised by retinal disease, and directly deliver a light stimulus to the inner retina in order to restore vision. Not a perfect solution. Ganglion cells are in an odd topography, although you have lots of pixels, hundreds of thousands of pixels to work with, unlike a microelectrode array. You bypass the circuitry for light adaptation. One of the main functions of the retina is gain control, and now you're inserting your image at a stage further downstream than light adaptation, so you're going to have to carefully control your light stimulus to match the dynamic range of ganglion cells, and that hasn't been solved. But still, I think it's an interesting approach, and I think the message I want to leave you with is that these methods are providing a two-way optical communication with a living primate retina, and as many of you know, this same technology is being used to try to understand how the brain works. In our case, we're looking at a very simple part of the central nervous system, the retina, but the same basic concept can be used, as you know, in the brain. And I think we're really at the beginning of an exciting revolution in which we're going to be able to have a dialogue with neurons. And if we really ultimately want to figure out how the brain works or the retina works or nervous system in general works, we're going to need some way not only of recording relatively non-invasively from many cells at a time, but also stimulating them too. So we have this two-way communication. So I think the future for this technology is very bright. I don't think Matteo would disagree with that comment in my talk. And maybe some of the others of you wouldn't either. But if you do, let me know. So I think that's what some of the things that adaptive optics has to offer the neuroscience, field of neuroscience going forward. I didn't tell you much about its use in studying retinal disease, but that's also moving along in exciting ways, I think. And so thank you. I'll take some questions if there's any time.